Hello, my name is Hatem and I'll first be presenting the molecular tools in oil palm research that is used in FGV. Basically, these tools is conducted to produce a hybrid crossing between parental oil palm plants of Dura and Piscifera to obtain Tenera oil palm plants that bear the most. These tools also analyze and profile the hybrid oil palm seedling based on the parental genome profiles. This process is very fast and time saving compared to the conventional method. One of the tools is polymerase chain reaction technology, where DNA of the seedling produced are analyzed and compared to paternal DNA, which is a concept of DNA analysis which is mentioned by Dr. Azinuddin as a concept in fluorescence science. PCR mimics the biochemical process of DNA replication, such as the process uses DNA template, denaturation, and primer annealing and primer extension. As you can see, this process is very similar to biochemical DNA replication. This process uses a very specific enzyme called tag polymerase to make new strand based on the DNA template. Another method used is next generation sequencing, which is a technique in which millions of nucleotides are deciphered at the same time for massively parallel sequencing for DNA, RNA, and methylation sequencing. This methodology consists of 3 steps. One, library preparation, where DNA libraries are created using random DNA fragments and followed by ligation via custom linkers. Number 2, amplification, where the libraries are amplified. And number 3, sequencing, where DNA is finally sequenced. Second principle of biochemistry used in FGV is plant tissue cultures. First, I want to present the reason why it is used and its objective. Reason number 1, seed germination rate of oil palm is very poor and take a longer period of germination under natural condition. Reason number 2, vegetative propagation is impossible for oil palm as it does not produce auxiliary shoots. So, plant tissue culture is applied to produce exact copies of oil palm plants with desired traits for large-scale planting and to reduce chances of transmitting disease, pests, and pathogen as plants are produced in sterile condition. Next, I want to present the process of orchid selection. Basically, orchid is the parent plant of clones, and only the best individuals for best crosses are selected. These individuals possess a combination of desired trait, and this orchid must have higher than 32% of oil to bunch ratios and high bunch numbers.
Next, I will continue with cloning through somatic embryogenesis. Somatic embryogenesis is the commonly employed tissue culture approach for the micropropagation of oil palm, and it helps to maintain the high-grade performance of seedling. And there are few steps involved in this method. First is induction of colors. In a medium containing alcim, induction of colors from explant of orchid, and this step takes around 3 to 12 months to complete. And next is formation of embryogenic colors. This step also needs a medium containing alcim, and proliferation of embryogenic colors happen in 6 to 15 months. Next step is maturation of somatic embryos. In a medium with reduced alcim concentration, progression of embryogenesis happens in 30 months. And the last step is plantlet regeneration. Regeneration of shoots spans around 16 to 36 months. On the other hand, process of fruiting needs around 2 months. Next, I would like to discuss about ramex. Ramex is a clonal plant which produces exhibit vigorous and uniform growth with very low somaclonal abnormalities, which is less than 5%. What is somaclonal abnormalities? Somaclonal abnormalities are in the form of manlet fruit that characterized by the presence of extra carpels, resulting in bunch abortion and sterility. Now, I would like to discuss about Karma test. The purpose of Karma test is to limit the impact of manlet abnormality. These tests also play a very important role in detection of affected plantlet before they are being planted in the field. In addition, karma tests also can avoid wastages of labor, plantation area, and resources to mallet palm. Moreover, it also helps to avoid yield reduction. There are a few steps that involve in karma test. First is with the use of a collection kit, a leaf or plant is taken, and next, the leaf DNA is extracted and treated with bisulfate. After that, the bisulfate will convert the cytosine into uracil but leave 5-methyl cytosine intact. Lastly, methylation status of karma is detected with PCR assay. Methylation status is very important because loss of karma methylation will cause mallet abnormality.
I continue to explain on the beneficial microbes for crop protection. The most common pest and disease of oil palm are Ganoderma species, which can cause basal stem rot disease that can lead to reduction of oil palm yields. Second is Rhinoceros beetle, which can cause damage to the leaves of the plant. Next is bagworms, which can reduce the yield of oil palm by causing 50% devolution of oil palm trees. In order to overcome these problems, FGF have introduced biocontrol agents which are Trichoderma aspergillum M103 and Metarhizium anisophile. Both of these are environmental friendly. Trichoderma aspergillum M103 are able to produce biological compounds that are toxic to the pathogen like Ganoderma boninans. It also helps to improve plant vigor and able to produce cellulase, lignase and amylase enzyme. While Metarhizium anisophile is known as green Mascardium fungi, which have been proven to be highly pathogenic to arthropods like Rhinoceros beetle. The infectious is easily recognized a few days after that. FGF has introduced Palmagro biofertilizer to enhance the plant growth. This product contains avascular mycorrhizae fungus that has a symbiosis relationship with the plant roots, where the plant provides carbohydrate to the fungus and the fungus will provide nutrient to the plants. Besides, it also used to control Ganoderma. Biological nitrogen fixing microbes plays important role in plant growth, as it helps to improve the soil biological activities and remove certain plant pathogens. They have also established bioaugmentation using beneficial microbes through their R&D initiative. Bioaugmentation is addition of archaea or bacteria culture to speed up the rate of degradation of a contaminant. Bioaugmentation is used to compost empty fruit bunch of palm oil, where the oil palm waste is regulated into composite and then turned into organic fertilizer. This can increase the crop productivity and improve soil health. Lastly, palm oil mild effluent is a thick brownish liquid that contains high concentration of total solid, oil, chemical oxygen demand and biological oxygen demand. So, bioaugmentation is an effective tool for the treatment of wastewater generated from palm oil mining activities. That's all from us. Thank you.